Hello, everyone. Thank you for the invitation to give this talk. I will talk about some of the work in my laboratory. My laboratory at the University of Maryland School of Medicine works on Gaucher disease. This is a lysosomal storage disease. It is recessive, and it is caused by mutations in glucocerebrosidase. And as a consequence, there is an accumulation of glucosyl sphingolipids. There are three forms of the disease, roughly. Type 1, which is a mild form. There is type 2, which is fatal. There is neurodegeneration, and children die before age 2. And then there is a chronic form, type 3, which also affects the brain, and there is neurodegeneration. The symptoms are increased size of liver and spleen, and bone marrow has problems. There is Gaucher macrophages that lodge in the spleen, in the liver, in the bone marrow. And these Gaucher macrophages are engorged with lipids that cannot be digested. And then sometimes in the lysosome, shown here, there is accumulation of crystals of glucosylceramide. There are also problems with the skeleton. People who have type 1, even the mild form, have problems with osteoporosis, bone pain, necrosis. And in type 2 and type 3, the skeletal problems are even worse. So the enzyme that is mutated, glucocerebrosidase, cannot break down this glucosidic lipid here, and as a consequence, glucosylceramide accumulates, and there is also an accumulation of glucosylsphingosine, which is generated by deacylation from glucosylceramide by an enzyme called acid ceramidase, another lysosomal enzyme. These are the genotypes that we have studied in my laboratory. We have made induced pluripotent stem cells. I don't have to say very much about induced pluripotent stem cells. We have reprogrammed fibroblasts from patients with type 1 or GD1, type 2 or GD2, and type 3 or GD3. And these are the genotypes. As I said before, in type 1, this is a mild mutation, and type 2 and type 3 are very severe mutations. The enzyme is made on the rough endoplasmic reticulum. It is transported through the ER and the Golgi, and then it goes to the lysosome, and it is transported by a chaperone called LIM2. There are more than 300 mutations in glucocerebrosidase, and some of these are listed here. And in type 1, which is the mildest form of the disease, the predominant mutation is N370S, and it affects primarily Ashkenazi Jews. And the frequency of this mutation is about 70%. And then L44P is a more severe mutation, and anybody who has L44P in Gaucher disease then has at least type 3, if not type 2 disease. So these are some of the genotypes, and as I said before, in addition to all the hematologic and skeletal abnormalities, there is neurodegeneration, severe neurodegeneration, and this is maybe 10–20% of cases. So the treatments for Gaucher disease have been around for about 2 decades. The most common one until recently is enzyme replacement therapy, which consists of biweekly infusions of a recombinant enzyme. And there are also chaperones that stabilize misfolded enzyme, and these are going to become probably more important in the future. And then there is substrate reduction therapy. Substrate reduction therapy by glucosylceramide synthase inhibitors, and perhaps in the future acid ceramidase inhibitors. This enzyme will synthesize from glucose and ceramide, glucosylceramide, and there is one that has been approved by the FDA. It is elyglustat, and it is used to treat type 1 patients with Gaucher disease. And there are other inhibitors. Elyglustat doesn't cross the blood-brain barrier, unfortunately, And there are other ones that are also glucosylceramide synthase inhibitors that can cross the blood-brain barrier. And there is a clinical trial for type 3 Gaucher disease, the chronic form, and also for GBA1 associated Parkinson's disease. Then in the past, bone marrow transplantation for type 1 has been tried, but this is problematic because it is quite dangerous. One has to ablate the bone marrow. And more recently, there are papers published about gene therapy, at least in mice for now, and they use adeno-associated viruses. So in my laboratory, like in many other laboratories, we have used reprogramming technology, and we have reprogrammed Gaucher fibroblasts and also normal fibroblasts into iPSCs, and then we differentiated them into all these cell types which are affected in Gaucher disease. We have made macrophages, neurons, osteoblasts, hematopoietic progenitor cells. So what we have learned in the last few years, and we have published a few papers on this, is that like everybody else knows, there are problems in the lysosome. There is a problem with lysosomal biogenesis, and there is a problem with phagocytosis. These autophagosomes cannot fuse with the lysosomes very well, and then there is accumulation of the autophagosomes. There is an autophagy block and lysosomal depletion because mTOR phosphorylates devel, which is then degraded, and devel is a transcription factor that is required for lysosomal biogenesis and induction of genes required for autophagy. But what we have learned from our work is that there is another aspect that has not been very well studied until now, and that is that the mutant glucocerebrosidase causes abnormal development in several types of cells. We have found abnormal hematopoietic development, which recapitulates the anemias seen in patients. We have seen defective osteoblast differentiation, which recapitulates the osteoporosis and skeletal deformities and problems in type 2 and type 3. And then we have seen impaired neuronal development. And this has not been described before. And what we showed, we published this work, is that there is a downregulation of the Wnt pathway, and Wnt regulates anterior-posterior development, and there is a defect that causes a loss of dopaminergic progenitors and also loss of hindbrain progenitors. So I'm going to focus the rest of my talk on the effects of these mutations in neurons in Gaucher disease. And as I said before, the mutant GCase interferes with lysosomal biogenesis and autophagy through effects on devel. There is this molecule here, mTOR, which I will talk more about later on, that constitutively phosphorylates devel, and the phosphorylated devel transcription factor cannot go to the nucleus where it activates the lysosomal and autophagy genes. And so to study the problem, we considered that that the problem was caused mostly by a problem in sphingolipid metabolism, that there was a sphingolipid imbalance. There are other causes, there are other effects of GCase mutations, and there are important groups that are working on gain of function. But all the phenotypes that we found in our lab and that I'm going to talk about today are caused by loss of function. That is, the enzyme is not working, and the consequence is that sphingolipids, glucosyl sphingolipids, cannot be hydrolyzed and they accumulate. So what I'm showing here at the left is that after mass spectrometry, we found, like many other investigators, that in GD2 and GD3 and GD1, there is an increase, shown here in the blue bars, an increase in glucosylceramide. If we look at the right, there is an increase also in glucosylsphingosine, but the difference between glucosylceramide here on the left and glucosylsphingosine accumulation at the right is that while the glucosylceramide accumulation in the neuronal progenitors that we assay by mass spectrometry have an increase of maybe 2 or 3-fold, but here we have an increase of 100-fold in glucosylsphingosine. And this recapitulates what is seen in the brains of GD2 patients. These brains have up to 1,000-fold accumulation of glucosylsphingosine. And there is discussion in the field, what are the offending, what are the deleterious lipids? Is it glucosylceramide? Is it glucosylsphingosine? Is it both? Is it others? And what my lab is going to work on is on the possibility that it is glucosylsphingosine, and I will talk about today. So how is glucosylsphingosine generated? And these are the structures of glucosylceramide. There is a fatty acid moiety, which is hydrolyzed by acid ceramidase, and this is the structure of glucosylsphingosine. So we are going to focus on this complex, the mTOR-C1 complex. mTOR kinase is regulated by many proteins. There are about 30 proteins in this mTOR-C1 complex. And it is a major regulator of anabolism and catabolism. And it is regulated, it receives signals, it responds to nutrients and to growth factors. And I'm going to show you today that it responds to elevated glucosylsphingolipids. And when this is active, then mTOR is phosphorylated. It goes on to phosphorylate other substrates, including devel, and this is important for the autophagy lysosomal pathway, and this devel will undergo proteasomal degradation. So how does this sphingolipid imbalance cause lysosomal dysfunction? How do we go from here, from a deficiency inside the lysosome to a deficiency in an important enzyme to problems with lysosomes? Well, we are going to show data showing that when there is GCase deficiency, there is an increase in glucosylsphingolipids, and this mTOR complex, mTOR-C1, is hyperactivated, leading to lysosomal depletion. And here, this is a Western blot and also image analysis showing that this phosphorylated mTOR is elevated in 2 different patient neurons, which we call GD2B and GD2A here. And here, if we look at the images, we see that there is an increase in the stain of a substrate of mTOR-C1 S6. And in GD2, there is more green fluorescence because there is an increase in phospho S6. And the opposite is true for LAMP1. Here we see these red dots, which are lysosomes stained by LAMP1. And there are a lot of lysosomes stained in wild type cells, but there is a decrease, an important decrease in the GD2 cells. So this is what the lysosomal depletion, what we call lysosomal depletion. There are fewer lysosomes. And we have done this also with lysotracker, which measures, which is responsive to pH. So this is image analysis of neuronal progenitor cells in this slide, and in the next slide, I will show you that we have similar results in differentiated cortical neurons. So here we have lysotracker staining of wild type and GD2 cells, and there is a decreased level of lysotracker here. And when we look at the substrate of mTOR, phospho S6, there is an increase in phospho S6, shown here by the black bar here. And then when we inhibit mTOR with this compound called INC128, we see that we can rescue the lysotracker staining, and here we can decrease mTOR hyperphosphorylation. So when with mTOR inhibitors, this complex is inhibited, we have lysosomal rescue. The same is true with differentiated neurons. This is again lysotracker. We see that in the GD2, there is low lysotracker staining, and when we give the mTOR inhibitor INC128, there is increased signal. And then the reversal is true, the reciprocal is true when we look at phosphorus 6. Here, there is a lot of phosphorus 6 in the untreated GD2 cells, but we can decrease it substantially when we use the inhibitor. This is shown quantitated here. When one goes up, the other goes down. And this is a recurring theme in my talk today. And when we look at by Western blot with LAMP1 now, we can see that in the GD2 cells, there is a decrease in LAMP1, but we can rescue it with these two inhibitors of mTOR, INC128 and TORIN1. These are catalytic inhibitors of mTOR, and this is quantitated here. So when we look at the structure of glucosylceramide and glucosylsphingosine, we can see readily that glucosylceramide is hydrophobic. It has this fatty acid moiety that is not present in glucosylsphingosine, and for this reason, this is a very hydrophobic molecule, and this is an amphipathic molecule. There is debate in the field which ones are the pathogenic lipids, and the answer is likely both. But they are probably going to work in different ways because they have probably different locations in the cell. And also, it depends on the cell type. Glucosylceramide is very problematic in Gaucher macrophages because the Gaucher macrophages ingest phagocyte red blood cells, and these red blood cells have glucosylceramide on their surface, and if the enzyme is not very active, then it cannot digest that, and there is accumulation of undigested, incompletely digested red blood cells. And there is a lot of glucosylceramide which accumulates sometimes as crystals inside the macrophages. But I will argue that in the neurons, it's slightly different, and that this glucosylsphingosine is really very important. And even though there is up to 1,000-fold increase in glucosylsphingosine in type 2 brains from patients, some people believe that this is the culprit, but some people believe that it's still glucosylceramide. But I'm going to show you data that at least in our in vitro system, it is glucosylsphingosine. So when we looked at GD2 and we look at the substrates of GD2, phosphorus 6 and phosphor40BP1, there is an increase in GD2 compared to wild type cells. And there is also, in GD2 cells, there is lower LAMP1. And when we give these compounds, these drugs, they are substrate reduction therapy drugs, Genzyme 161, ebglustat, also known as venglustat, which is now in clinical trials, and eliglustat, which has been approved by the FDA for type 1 Gaucher disease, there is a decrease in the substrate of mTOR, S6, phosphorus 6, and 4-BP1, and there is a rescue of the lysosomes. And this is again quantitated here. We can decrease, of course, with the genzyme ebglustat and eliglustat, we can decrease the phosphorylation levels, and then the reciprocal is true for the lysosomes, we can increase them. So eliglustat unfortunately doesn't cross the blood-brain barrier, but Genzyme 161 and venglustat, ebglustat can. And ebglustat is in clinical trials, as I said before, for type 3 Gaucher disease, but also for GBA1 associated Parkinson's disease and this implies that a problem in GBA1 associated Parkinson's disease is an elevation of glucose sphingolipids. Now, this elevation is likely to be very small in PD patients, Parkinson's disease, in heterozygotes, because first of all, this is a recessive enzyme, a recessive gene, and second, there is very little accumulation of lipids, and some people in the field are arguing that there is no accumulation of glucosylsphingolipids. But the argument is that over a period of years, over the period that it takes for a person to develop Parkinson's disease, these small accumulations every day are likely to do something. Hard to prove, but I believe that this is the case. So I will show evidence from our laboratory, from our in vitro system, that elevated glucosylsphingosine may be responsible for mTOR activation and lysosomal dysfunction. So one of the things we did here is mass spectrometry analysis showing that in these two subclones of one GD2 patient, two subclones of another GD2 patient, and a clone of GD3, if you look at the bars, the black bars, there is a very large increase. There is here in this case, 60, 70-fold increase in the level of glucosylsphingosine. And this is also true, although to a lesser extent, in GD3. And when we inhibit this enzyme, acid ceramidase, with this compound, carmo4, then we bring down the levels of glucosylsphingosine in every case. So what happens is that when there is mutant GCase, there is an elevation of glucosylceramide inside the lysosome. This is a lysosomal enzyme, acid ceramidase. It will cause the appearance of glucosylsphingosine. It goes to the cytoplasm, and in the cytoplasm, it will activate mTOR. But when we inhibit it with this compound, carmo4, then we block, to a certain extent, this pathway. There is a lower level of glucosylsphingosine in the cytoplasm, and then there is lower mTOR hyperactivation, and then we can rescue the lysosomal phenotype. So this is image analysis and also Western blot analysis showing what happens when we give carmo4 to differentiated cortical neurons. So in wild-type cells, we have a certain amount of constitutive phosphorylation of the substrate of mTOR PS6, but it is elevated. This is the green fluorescence here in GD2. But when we give carmo4, then this inhibitor brings down the activity of mTOR. And the opposite is true for the lysosomes. When we use lysotracker, which stains according to pH, lysosomes and endosomes that are acidic, we have low levels of red dots here of lysotracker stain. But when we give carmo4, we can rescue that, and this is again shown here. We can see that with carmo4, we can rescue the lysotracker counts, and then we can bring down hyperphosphorylation. And here in this Western blot, we can see that there is an elevation of phosphorus 6 and 4 EBP1, of phosphor 4 EBP1, but when we give carmo4, we bring it down. And then in this Western blot, we show that there is, in GD2, there is less LAMP1 intensity of this band in this Western blot, but when we give carmo4, we can rescue it. And this is what I just described. So what is happening is that in the lysosomes, with this enzyme, ceramidase, under normal circumstances with wild-type cells, this enzyme will hydrolyze glucosylceramide to glucosylsphingosine and fatty acid, but there is virtually no detectable level of glucosylsphingosine in normal cells. This is not the case in Gaucher disease. There is quite a bit. And it is not that this enzyme is elevated. It is that it really works over time. And then this very high level of glucosylceramide is converted to a very high level of glucosylsphingosine, which is shown here. And then when we give the inhibitor carmofur, then by inhibiting this enzyme, we can rescue the phenotype, the mTOR phenotype and the LAMP1 phenotype. So where do we stand with acid ceramidase inhibitors? Well, carmofur is toxic. It has been used in Japan to treat some cancers, but we need a lot better ones. And now people are, investigators are trying to generate new ones because this is going to be an important enzyme for treatment. Okay, so let's move on here. So again, glucosylsphingosine is an amphipathic water-soluble glucosylsphingolipid. It doesn't have a fatty acid moiety, which has been hydrolyzed. And it has this amino group, which is protonated in the acid pH of the lysosome. It is hydrophilic. And then when it moves to the cytoplasm, it is still partially positively charged, and it is still amphipathic, and it is in the same compartment in the cytoplasm where mTOR is. So we can imagine a scenario where we can activate mTOR C1. Whether it's direct activation or indirect activation, we don't know. There are, as I said, almost 30 proteins in mTOR C1, and we don't know how it works. So we wanted to look a little bit more, obtain more evidence for or against this role of glucosylsphingosine in pathogenesis, in the neuronal phenotype. And what we did is we decided to treat the cells with exogenous glucosylsphingosine. Now, we are not sure that when we treat cells with glucosylsphingosine, it will go to the same compartment inside the cells as the endogenous glucosylsphingosine, but we have to start somewhere. So what we did is we used increasing concentrations of glucosylsphingosine, and they do activate in a dose-dependent manner mTOR, as shown by the increase in this green fluorescence. And again, the opposite is true. One goes up, the other, LAMP1, goes down. And when we did the Western blot, again, these are, I have to emphasize that what we are treating here are wild type cells. We are not treating the Gaucher cells. So when we treat the wild type cells with increasing concentrations of glucosylsphingosine, we see an increase in phospho-mTOR. And when we compare it with GD2 here, it is a substantial increase. And this is a quantitation. We now submitted a paper on all this work. So here in this cartoon, glucosylsphingosine will activate the mTOR C1 complex, and there will be lysosomal depletion through mTOR hyperactivity, which probably works through devel. So does mTOR mediate the effect of glucosylsphingosine? So what I'm implying here is that glucosylsphingosine causes lysosomal depletion and the problem in autophagy through mTOR. But is this true? If we treat the cells with glucosylsphingosine, what happens when we inhibit mTOR? Will there be lysosomal depletion or not? So here in this Western blot, what we show is that by treating with glucosylsphingosine in the last 3 lanes, first of all, there is an increase in mTOR. So glucosylsphingosine does activate mTOR, as shown here by increased phosphorylation of pMTOR and PS6, and there is a decrease in LAMP1. And when we incubate with 2 mTOR inhibitors, either INC128 or TORIN1, then what we see is that, of course, if we are inhibiting mTOR, these bands are going to be less visible here. We wipe out pMTOR and we wipe out PS6, but we also increase LAMP1 levels here. So what we are showing here is that yes, if we use mTOR inhibitors, we block the action of exogenous glucosylsphingosine. Whether it is direct or not, we don't know. So that was the last of my slides, and I'm going to give you a summary. So we showed that glucosylsphingosine treatment of wild-type neuronal cells recapitulates the phenotype that we see in Gaucher disease neuronal cells. These deleterious effects of glucosylsphingosine on lysosomes are blocked by inhibition of either mTOR or acid ceramidase. And I am going to postulate that mTORC1 is a sensor of glucosylsphingosine. Of course, we don't have biophysical evidence that it interacts with any of the mTORC1 proteins, but at least we know that mTORC1 is responsive, directly or indirectly. And we are now beginning work to see how this works, to look at the mechanism. So I have shown you that if we use glucosylceramide synthase inhibitors, we decrease the levels of both glucosylceramide and glucosylsphingosine, and we see a rescue of the phenotype, but we don't know which one is the offending lipid in these neurons, because glucosylceramide will decrease the levels of both. But if we block glucosylceramide, we ask the question, is this sufficient to rescue the phenotype? And the answer from all the data that I show you is yes. So this is the last enzyme before the appearance of the last enzyme involved in the synthesis of this lipid. So it might be more effective to use an acid ceramidase inhibitor than a global glucosylceramide synthase inhibitor. And glucosylceramide is important in the cells. We cannot just inhibit it completely because it will have some side effects. But glucosylsphingosine is actually almost not detectable in normal cells. So inhibiting it is not going to have probably as a deleterious effect. Although a knockout of acid ceramidase, there are mice that there is a human disease and there is an accumulation of ceramide and there are problems. However, by regulating the dose, we may find some compromise that will be therapeutically effective. So I have shown you that if we inhibit mTOR with mTOR inhibitors, then we can also block the abnormal phenotype, the lysosomal depletion. And here I put this, we are doing some work with AMP activators like metformin, and it would be nice if metformin would be helpful in GD, in Gaucher disease, in neuropathic Gaucher disease. Metformin is used by millions of people who have diabetes, and there might be already epidemiological data comparing the incidence of Gaucher disease phenotypes, clinical phenotypes, in people who take this drug compared to other people. So this is a summary then, and I would like to thank the people who did the work. All the data that I showed you today was done by Manza Srikant, a graduate student in my lab who just completed her PhD and is staying in the lab to finish up a few things, but then she's going to go for a postdoc. Ola Awad made the initial observations that in Gaucher disease, there is wind down regulation and mTOR hyperactivation. And then these are people who are working on other projects in my lab. Manoj Kumar is working on GBA1 associated Parkinson's disease caused by mutations in GBA in heterozygote patients. And Afsun and Jackie are working with macrophages. And our collaborators are Jace Jones and Maurice Kane, who did the mass spectrometry analysis. And these are our collaborators at Harvard and Germany, Ole Isaacson, Penelope Howard, and Michelle Adelaide, who are working with us on GBA1 associated Parkinson's disease. I did not talk about with you today. We have some nice data coming along, and I'm really very lucky to be collaborating with them. And I will, the only thing is that these are my funding sources. Unfortunately, I cannot take questions from you, but thank you very much.